Welcome student in the last lecture of our SWAM course, Anatomy of Flowering Plant. And in this course, we will try to discuss about some anatomical structures we have not discussed before. Like we have talked about stem, leaves, root, about secondary growth as well as enameller structure, different adaptations. But still we haven't discussed the anatomy of reproductive structures, like we have flowers, we have seeds. Okay, so we will try to discuss those structures in this lecture, and also in the last we will try to find out some application of plant anatomy. Okay, so starting with the anatomical structure of the reproductive parts which are present in a plant. So definitely the reproductive structure is the flower. Okay, now before we move on with anatomy, let's just in brief try to have its morphological description. So in flower you will find different whorls or different parts of a flower. You will see that flower will have a stalk. Okay, it will have a base known as receptacle. Okay, thalamus, on which all that floral whorls will be present. Now you will see the first whorl will be known as sepals or calyx. Okay, so calyx will be made up of sepals. Different parts of that calyx is known as sepals. So basically they will be green in color, although a lot of modifications are also there. Now after this whorl, you will see presence of colored petals. Okay, they are known as corolla. Okay, so the next whorl is corolla. And it is made up of petals. The number of petals can vary. Okay, similarly the number of sepals can also vary. They could be fused, they could be free. So a lot of different morphological types could be present in all those different parts. Then after petals you will have the most important parts of the flower, which are the reproductive parts. These sepals or petals, actually they are not taking part in reproduction, but they are helping in reproduction. And the most important parts which are actually taking part are the androecium and gynoecium. You have androecium, that is the male part, and gynoecium, that is the female part. Okay. Now coming to androecium, they will be made up of stamens. Okay, so group of stamens are known as androecium. If the number of stamens can vary. Now each stamen, when we talk about stamen, okay, these are all stamen. These are the third whorl. Okay, these are all stamens. So in each stamen you will have anther and filament. So this is the stamen. Okay, and you will have anther, the lobe structure, which will be found at the tip of filament like structure. This is the filament and this is the anther. So this is the male part, and this anther will contain pollen grains. So we will talk about this anther structure in detail, because that is the one of the most important part which is giving rise to the male organ, which is giving rise to male gamete or the pollen grains, which will take part in fertilization, and this will contain pollen grains. Okay, now coming to the female part, that is the gynoecium. Now this will be made up of carpels. Now you can have one carpel, you can have several carpels. Okay, now each carpel will have basically three parts. They could be fused, they could be united, so different orientations could be there. Now it will have stigma, style, and ovary. So inside this ovary you will have ovules. So ovules are the important structure which will give rise to female gamete. So in pollen grains you will have male gamete, and in ovules you will have female gamete, and during pollination that pollen grain will land on the stigma, pollen tube will form, and it will go and it will enter inside the ovule. Inside the ovule you will have a female gamete, and pollen grain male gamete will come and they will unite to give rise to zygote, and finally embryo will be formed, and then the entire plant. Although we are not going in that detail about how all these processes will occur, but we will try to understand the anatomical structures of these part. So coming to the first important structure, that is the androecium, that is group of stamens. In that also we have discussed that you have anther lobed structure and which is connected by this filament. Now anther could be definitely, see there could be number of lobes can vary, okay, but generally we see an anther which are bilobe, even they could have one lobe also. Generally the anther has two lobes, okay, and it has a filament. Now these two lobes, see when you will cut the section of this anther, actually inside this anther only you have pollen grains. So you can imagine that anther lobes, inside that you will have a bag or sac like structure in which lot of pollen grains will be formed. Now however their formation is also a thing to be discussed, but in anatomy we are not going to discuss all those things. Now once you will cut the section of this anther, you will see all the layers which are present around it, okay. This is the longitudinal section, okay, but this is the transverse section. So when you are cutting the section, you can see that how different layers are arranged, and inside this also what is present. Now generally this anther is two lobe, but in each lobe also you can have two cavities, okay. So in each lobe you are having two cavity. You can see here that this is one lobe and the other one is, see this one is the entire lobe, okay, and this one is the entire lobe, but in this also you will have two chambers. In this also you will have two chambers. So when you will cut and see the transverse section, you will see that basically four chambers you will see, okay, four chambers. So in this four chambers, inside this you will see four sac-like structures or bag-like structures in which pollen grains will be present, okay. So you can see here that this is the pollen sac, okay, and they have lot of walls also. Now this is known as microsporangia. Sporangia, okay. This whole structure, this whole bag-like structure, four structures will be there, sac-like structure. This is known as microsporangia, okay, and they will be surrounded by wall layers. Lot of wall layers will be there. So all these wall layers are important, and we will see what is their composition. And inside this microsporangia, you will have pollen grain, and this type of anther is known as tetrasporangia. That means four microsporangia is present. See, this is the most common version which is present, but the variations are definitely there. Now we will try to understand the structure of this tetrasporangiate anther, okay. Now definitely it will be surrounded by epidermis. See, I will draw detail of only one side, and you can understand how the other sides are also oriented. It will show four microsporangia here, okay. So it will have four wall layers. So outer wall layer is definitely the epidermis. The epidermis is the wall layer which is present in all plant organs and surroundings, okay. So everywhere it will be there. Now this will be followed by another layer which will be actually some radially elongated cells. Okay, then this will be followed by two or three middle layers. And this microsporangia will be finally surrounded by a layer of cell which will also be slightly bigger in size, and I will tell you what are their names. Okay, and inside this you will see lot of microspores or pollen grains. So in this way, these wall layers will be present around each micro sporangia. So now this is epidermis. Now the next radially elongated cell is known as endothecium. Now endothecium is very important. It has fibrous thickening. It has fibrous thickening, and this endothecium actually helps in rupture of the anther wall, helps in rupture of the anther wall. Okay. Now, because of this fibrous layer, because this fibrous layer is actually hygroscopic in nature, so it will absorb moisture and it will swell, and at these points, which is known as stomium, the fibrous thickening will be very less. Okay, so they will be the delicate areas, and well, all the other cells will swell, these delicate areas will open. Okay, so this is the stomia through which this anther wall gets ruptured. Now, after endothecium, you have 2-3 layers of middle layers. Now, middle layers, when the anther will mature and when it is about to rupture, these middle layers will actually get degraded and they will be lost. Now, the next important layer which is actually surrounding this microsporangia is tapetum. Okay, this tapetum is very important. It provides nourishment to the growing pollen grains. Okay, now this tapetum serves lot of function. This tapetum is also multinucleate. So this tapetum has a lot of roles. First of all, it is providing nourishment, it is providing food to the growing pollen grains, because inside this microsporangia, pollen grain will develop. Now, how this all will develop, there is a entire process that in between you will have, first of all, sporogenous tissue, and then they will give rise to microspore mother cell, and this mother cell will give rise to by meiosis. Okay, so that all, if you don't know, you can go back and try to find out, because in this class I won't be able to explain all those things. Meiosis is a reduction division by which 4 microspores are formed, and then these microspores will develop into pollen grain. Okay, so when this pollen grain is growing and developing, microspores are dividing. Okay, so they all need food, so that is provided through tapetum. Now, tapetum is multinucleate because lot of function it has to do, so extra work it has to do. That's why its genetic content is also very high. It also contributes many of the components which are useful in formation of pollen grain. Okay, we will discuss about the structure of pollen grain. There we will find out that how different layers are there and how tapetum is also contributing. So in between you will have sterile tissue which will be connective. Okay, which is known as connective. So between these fertile tissues, all these microsporangia, you will have a connective layer. So that will be sterile in nature. So this is a structure. Now there you can, this is the microscopic slides pictures. You can see here that all wall layers are there. This is the endothecium. Okay, you have microspores inside it. Okay, tapetum is there, then epidermis is there, then middle layer is there. Okay, this is all tapetum, and this is all microspores or pollen grains. So this is the pollen sac. This is also known as microsporangia, is also known as pollen sac. Now, when these pollen grains have completely developed, they will actually they have to be released. Okay, so they are released at this point, which is known as stomium. So I have told you that endothecium will help in that. The stomium will have a delicate wall as compared to endothecium. Fibrous thickening will be less, so they will open up from there. Pollen grains, the matured pollen grains will come out. Now, when we talk about pollen grains, see, they are very important structures. The structure of individual pollen grain, actually, you will see that they will have an outer wall and an inner wall. Okay, so this pollen grain will have outer layer known as exine and the inner wall known as intine. Okay, and inside this they will be divided into two cells known as generative cell. Okay, and this is the vegetative cell, and it will have its own nucleus. This is generative cell nucleus, and this is the vegetative cell nucleus cell. Okay, so this generative cell will later give rise to two male gametes. So now here in the outer wall you will see that which is exine. You can see at certain points pores are there. So these are known as germ pores. Now this outer exine layer is actually very thick and very strong. Okay, it is made up of, exine is made up of a very strong material which is known as sporopollenin, which is considered to be one of the hardest material which is present on earth, and it is non-degradable. It is even preserved, because of which pollen grains can survive under harsh condition, and they could be preserved for long also because of the presence of that sporopollenin. Now tapetum is, as I have told you, that is it's a very important layer, and it's not only provide nourishment, but it also secretes many components which also becomes part of this pollen grain. So sporopollenin is also secreted, and some other important Ubish bodies, these structures are also secreted from tapetum, which become part of the outer wall of this exine. Now these pollen grains, not only they definitely they contribute in fertilization. These are the male reproductive parts, and they contain male gametes. It is also important that these pollen grains, lot of structures are there, means lot of variation in these structures are present on that pollen grain, and it is so highly diverse that an entire branch of study is devoted on studying the structure of these pollen grain, and this is known as palynology, means the study of pollen grains. You can see here a lot of pollen grains which are of different sizes, which are of different shapes. You can see round also, you can see a very small, bigger one, oval shape, elongated, flat. So different structures are there, and different sculpturing patterns are also observed on these pollen grains. You can see here, like this is the outer exine. Okay, in that outer exine, which is actually a very thick layer, lot of sculpturing patterns. Suppose this is the intine. Okay, and this is the exine layer. Okay, so in exine layer you can have a lot of ways by which this exine layer can be oriented. This can have some pits, it can have a slit like pores, it can have a star like appearance. So these pollen grains can have lot of pores, these germ pores. So germ pores shape can also vary, their slits can also vary. So lot of variation is there in these pollen grain, and they are studied under a different branch that is known as palynology. And this pollen grains can also be of taxonomic significance. You can identify the plant on the basis of pollen grains. Okay. So now coming to gynoecium, that is the female reproductive organ. So in this you will see carpels, it is made up of carpel. So each carpel, when you will analyze, it is made up of 3 parts. Okay, so this is the stigma, style and ovary. Now inside this ovary you will see ovules, okay. So ovules will contain the female reproductive part, okay. Now just we will try to, now one ovule could be present inside this ovary, or several ovules could be present, as you would have observed the seeds of the plant. See, seeds are modified ovule only, and the ovary is a modified fruit, like tomato fruit or any other fruit. Mango fruit has one ovule because you are getting one seed. Like in tomato you will find lot of seeds, so that means lot of ovules are there. Okay, so in that way you can analyze the fruits and you can find out that how many number of ovules are there and how they are also attached with the ovary. Now they are attached with the ovary through placenta, means placenta is a tissue through which this ovule is attached. Now suppose this is the ovary wall, okay, and through this placenta this ovule is attached. Now I am just drawing a single ovule structure to make you understand that how they will look like. So ovule will definitely be surrounded by wall layers. Okay, so this single ovule will be surrounded by integuments. Generally 2 integuments are there, outer integument and the inner integument. Okay, it will be attached with the stalk, which is known as funiculus. Okay, now this will be connected with the main body through a structure which is known as hilum. Then there will be an extension of this stalk to the entire body, which is known as raphe. Okay, now in this ovule you will have two ends. One is the chalazal end and one is the micropylar end. You will see that the integuments are actually leaving a pore on one side. They are not completely connected. So on one side there is a hole through which actually the pollen tube will enter. So this side is known as micropylar end and this pore is known as micropyle. Just opposite to this micropyle will be the chalazal end, okay, or chalaza. Now inside this there will be a tissue, okay, which is known as mucilus, and in this there will be the embryo sac. This I am drawing the structure in which a complete embryo sac has been developed. So this embryo sac will actually contain the female gamete. In this embryo sac also you will see structures, you will see cells, okay. So the 3 cells on the chalazal side will be known as antipodal, and the 3 cells which are present on the micropylar end, in which the central one is known as egg cell or female gamete. And the other 2 are known as synergids. Okay, so this is the organization, and in the center you will see 2 polar nuclei. So they will all participate in fertilization process. So male gamete and female gamete will fuse. One, see, pollen grain will fall on this stigma, okay, pollen grain is falling on this stigma. Then they are germinating with the help of germ pores. Means germ pores are the region through which the pollen tube will come out, because the entire exine is very tough, so it's very difficult to break the entire exine. So they will come out through germ pore, they will enter and they will reach the micropylar end. Okay, they will enter inside it, where the nucleus or the male gamete will be released. Okay, where you will have these cells. So they will, male gamete will fuse together, will fuse with the female gamete, and this will give rise to zygote. Similarly, the other male gamete will fuse with the polar nuclei and they will give rise to embryo endosperm. Okay, this will act as a source of nutrition. Now, after this zygote formation, definitely they will give rise to embryo and the entire seed formation will occur. Just in brief, we will try to understand how the seed will look like. See, you all have seen the seed of a dicot plant and a monocot plant. See, in a dicot, you will see, like you imagine pea seed. Okay, when you will split the seeds, you will see that two cotyledons are there. Cotyledons are there. Okay, so in this you will not see endosperm because endosperm has been completely used up in the development of the embryo. So here what you will see a small embryo like structure which will have two ends, which will have a hypocotyle, which will give rise to the radicle or the root part. Okay, and this will have the plumule. Okay, this will have the plumule. So this will give rise to shoot. Similarly, when you will observe the structure of monocot seed, like maize, if you will see, what you will see? See, in that case, you will see endosperm, that is endosperm is still present. Okay, you will see that endosperm is there. The seed is surrounded by a pericarp or seed coat. In this case also you have a seed coat. Okay, and in this you will have an aileron layer. This is very important layer. We have also studied that in aileron layer protein grains are there. Okay, layer. Okay, then this you will see scutellum, which is actually a single cotyledon. Okay, then you will see some plumule and radicle, which will be covered by a covering known as a coleoptile, and this is coleorhiza. So basically this is the embryo part. Okay, you will have plumule here and you will have radicle here. Right. Okay, so this is the structure of seed of monocot and dicot plant. Now in brief, we have tried to understood the anatomy of reproductive structures. Now coming to another important topic which I just want to discuss in last is the application of plant anatomy. See, we have studied about the different internal structures of plant tissues and organs. Now, this is definitely very important, but there are different areas where you can apply the knowledge of this anatomy. Particularly, see, you can apply in the knowledge of this anatomy, first of all, in understanding different branches of plant science. Now, we have discussed so many things. We have discussed that structure is always related to function. Okay, so when you understand the structure, you can also understand the function of that structure. So like for example, to understand the function of photosynthesis, you should understand the structural organization of leaves and how different tissues are involved in photosynthesis. Even inside the internal structures also, we have discussed like how chloroplast inside that, how different components are organized to perform photosynthesis. Then respiration also, how different tissues and internal organization is there. Now different tissues are involved in conduction of water, translocation of food particles, and then for support, for providing mechanical support, different tissues are involved, and how they are organized, how they are organized in different plants also, which are adapted to different environment. So it's important that you should understand the structure in order to understand the function. So the understanding of plant anatomy will help you to understand plant physiology. That means the different functions of the plant. Similarly, in plant ecology also, the knowledge will be of so much of importance. Plant ecology means how plants are adapted to different environments, how they are growing in different environments. See, unless and until you understand the structure, you cannot understand their function. And then in developmental biology or plant development, you can understand the developmental process. Okay, so all these branch of sciences, it's not limited to that. In different fields, you can apply different branches of science. You can apply this knowledge and you can better understand those things. Now, this plant anatomy is important in identification of plants and taxonomy. See, like you, many a times you need that the proper identification of plants should be done. Like if a person is a plant breeder, want to breed different types of plants, they are working on cytology, like cellular structures, means cellular organization and cellular functions. They are working on that. They are like working on foods of plant, on by like medicines, then furniture, things, various areas. When a person is working on and they require a particular plant, it's important that they should identify the plant. So, and second thing is that when a plant is identified, suppose you find a plant, okay, you don't know what is that plant and in which group it falls. So you will study that plant and you will try to analyze them and try to classify them. So that is taxonomy. So you can classify the plants also based on their anatomy. Previously, actually, a lot of morphological characters like vegetative characters, floral characters were generally used to classify them. It means flower structures, the arrangement of different floral parts, different leaf structures, and how they are organized. Morphological structures, they were used. But now anatomy is also serving a very important tool to identify those structures. Now there are various things in anatomy which you can use, like you can use trichome anatomy, trichome structure. Okay, trichome, different types of trichomes are present on different plants that we all have studied. So on that basis, you can identify plant, you can classify them also. You can study leaf anatomy, wood anatomy. Wood also we have studied before. Then different leaf anatomical structures are also different, like monocot leaf will have a different structure, dicot leaf will have a different structure, stem and root, they will also have a different structure. See, many of the differences are based on their anatomical feature only, like vascular bundle arranged in the form of ring, then monocot will have a different arrangement. Even within that also a lot of variations are found. Then leaf epidermis is also very important feature which could be used in differentiating. See, on leaf surface, epidermal cells are there, pavement cells are there. Okay, their arrangement is different, they are arranged in different orientation. Okay, so their size, shape and their different orientation could be observed. Similarly, stomata and guard cell as well as subsidiary cells, their arrangement could also vary. So their arrangement could also vary. That also we have studied, that there are different orientation, and depending on the plant type, these orientations could be found. So that could also be a basis for identification of plant and classification of plant. Similarly in vascular bundles also, you can have bundle sheath. Okay, that could be parenchymatous, that could be sclerenchymatous, and how they are arranged, how they are oriented, that could also be a basis for classification. Now, presence of cuticle also, presence of cuticle, waxes. Now, the thickness of the cuticle and many times the orientation of the cuticle and waxes also vary. See, that also we have discussed, that cuticle could be continuous, they could be arranged in a continuous layer or in some sort of granular form. some sort of needle-like structures, or some different structural modification could be there, and those could be found in different plant species. Similarly, many crystals are also found in different plants. Okay, so they could also be an important feature of taxonomy. Then you have calcium oxalate crystal. Okay, idioblast cells are there. Okay, lithocysts are there. Then cystoliths are there. Okay, oxalate and carbonate crystals are there. Okay, then silica bodies are there. So all these structures could be a feature of could be taken as a feature that could be used for identification and for classification. Similarly, you have palisade ratio. See, in leaf also you can have a different ratio of palisade layer. Okay, they can be 1 or 2 or several layers. So that could also be analyzed. Similarly, primary and secondary growth could be analyzed. Okay, how in secondary structures also lot of variation could be observed. Lot of anomalous structures are found that could also be observed to identify a plant and to classify them. Lot of ultra structures even in trachees and vessels. You can have pits, you can have secondary thickening, how sieve plates are there. Okay, sieve, their ultra structures will tell you how is the organizations of sieve plate, how pits are organized, and what type of secondary thickening is present. So see, this all vary depending on the plant type. So that could also be used. Second important application could be find out adulterants in crude drug. Now, medicinal plant, as you all know, that it's worldwide it is used, and generally they are used in dry form. Now, many times you would have seen dried bark or dried leaf, even in powder form also they are sold, or you get dried root is there. So you can bring it also, you can crush it also, or in dry powder form they could also be available. So adulterant addition of some other compound or addition of some other plant part is also a very common practice. So how to find out the purity, how to check the purity of the drug? See, one thing is that you can see morphologically you cannot check it. Okay, so next thing is that you can go for chemical analysis. Nowadays it is also becoming very common, and it is very important method to identify the chemicals of those compound, but it will definitely be it will be costly. Okay, and it is not applicable every time. So one thing is that you can identify the anatomical structures. See, in powdered form also, or in crude form, when the plant is completely dried, some of the anatomical structures are still present. Like suppose a cystolith is there. Okay, cystolith, okay, which contain crystals, they are preserved. Even the pits are preserved. You can say even the stomata, the structure and the organization of the subsidiary cells, they are all preserved. Starch grains are preserved, like wheat and starch grains are there. Some medicinal plants are having some starch grains. Many times wheat starch grain could also be mixed in that. So in that way you can find out the difference. Now it could also be used to identify wood. See, wood also we have discussed that different quality of wood is there based on presence of vessels or absence of vessels, how vessels are organized, whether they are scattered or grouped together, then presence of parenchymatous tissue attached with the vessels, then you have a pit. So all these factors could be useful in identifying the wood and finding out the quality of wood. Similarly, in archaeology also, these anatomy will be very important. You can identify the specimen based on their anatomical structures, particularly like wood or even pieces of charcoal. If they are there, you can identify on their perforation plate. These many structures are still preserved, anatomical structures are still preserved, and on that basis you can identify. Sometimes they are used in tissue culture technique also. You can use anatomy, like you can use the meristematic cells to culture. Okay, you can identify those meristematic cells and you can culture them for vegetative propagation, for producing clones of that plant. Okay, many times this meristem culture produces virus free plant, and it produces clone because it divides so fast that viruses don't get time to replicate. So you can have virus free plant when you are using meristem culture. And they can also be used in forensic investigation, means solving criminal cases with the help of science. In plant science, you have a plant anatomy which you can use to solve criminal cases, like suppose if some pieces of plant is present on the site of on the crime site of, or a person who is a suspect carries a piece of plant material, and you are suspecting that that person might be present on the crime scene. So you can analyze those small pieces of plant material. So I have told you there are different segments, even a small piece like cell wall fragment is there, some trichome is there, so that on that basis pollen grains are there. On that basis you can identify the plant, and you can identify whether the person was there. Similarly, food particles which are there in the stomach, so you can identify them. If they have those anatomical features still intact, you can find out what was the last meal. Similarly, you can find out if the body, how long the body would have been buried based on the fragments of plant parts which are there. Then if it has been shifted from one place to another, if it is carrying the fragment from one location to another location. So all those things like silica bodies, raphides, that all we have discussed, that these are crystals. These are crystals which like druce, so sclerids are there. So all these things could be identified, and on this basis plant could be identified. Okay, so this is some of the application which anatomy has, and I hope so that you all would have enjoyed learning anatomy, and I have tried my best to explain you the basic concept and make you understand the general anatomical structures and how you can differentiate and find out the anatomical structures of different plant types. And this, I think so, this will help you in understanding not only anatomy but also other subjects. You will better understand them, and you can even think of many other things exploring in plant anatomy, and you will try to do better in your life. And all the best for your future, and thank you for enjoying this course, and I hope you would have enjoyed it. Okay, so thank you.